postdoc from UK as a Newton Fellow, and then I joined Department of Biochemistry as a Ramanujan Fellow. Today I am going to talk about the topic of proteomic perspective on neurodegeneration, navigating molecular pathways in neurobiology. So, what are the neurodegenerative diseases? Yes? Those components includes neuron or the some parts of the like glial cell, oligodendrocyte, all those things. So there are some genes established for some of the diseases, like for Alzheimer's. We know that there is amyloid. So amyloid beta plaque or the overphosphorylation of tau and amyloid beta plaque accumulation. Then it includes a lot of diseases, like prion diseases are also part of neurodegeneration, Parkinson's. Then there are FTD, frontotemporal dementia, then ALS. So what is their hallmark of neurodegeneration? First thing is neuronal cell death or pathological protein aggregation, synaptic and neuronal network defects, aberrant proteosomesis, aberrant degradation of proteins, which is one of the most common example of this is, come on, APP. Then cytoskeletal abnormalities, altered energy hemostasis, DNA and RNA defects, inflammation and all. So the first thing is how. So what are the proteins that get misfolded and result into the neurodegeneration, which include amyloid beta, tau, prion, alpha synuclein and TBP43. So it's very easy, you can see there are monomers, then oligomers. So basically they accumulate and they form the fibril and ultimately the plaque. So based on the clinical features, neurodegenerative diseases are classified into two parts: cognitive predominant disorder and movement predominant disorders. So the cognitive, where the cognition, the mental part is involved, and the movement which affect the muscles, so the movement is affecting. The cognitive part predominant includes the Alzheimer's, FTD, the frontotemporal dementia, the prion disease. The movement part include Parkinson's, atypical Parkinsonism, motor neuron, ataxia and chorea. So we have already discussed the misfolded proteins are amyloid, tau, prion, and the location of the accumulation could be neuronal, astrocytic, and it could be oligodendroglial as well. So pathologically, Alzheimer disease is not, so we say Alzheimer disease as the mix. It includes both APP and the tau. So it's mixed pathology. So what happens initially, there are diffuse plaques of APP. Then as they get progress, they get denser in the center and diffuse on the sides. This denser part further attract the tau accumulation. This is histologically, and then this tau accumulation, because of the over phosphorylation, it leads to it pathological histologically it looks like accumulation of the tau in the neuron bodies. Then this was because of amyloid beta and tau accumulation, so mixed. Now there another protein which leads to NDD is alpha synuclein, and it includes Parkinson's and multiple system atrophies. So typical feature of Parkinson is Lewy body, and it could accumulate either in the so this is the immunohistochemistry of the protein alpha synuclein, and this is the pictorial cartoon so how typically it could be accumulated in the cytoplasmic part or in the fibrous of the neurons, and in the multiple system atrophy in the glial it could be either arranged in the sickle shape around the nucleus, or it could be in the nuclear. So this is the nucleus of the neuron, and this brown with the blue arrow is the inclusion body. Then these are these are various diseases leading to the neurodegeneration, and 3R, it could be because of the 3R or 4R tau. What is 3R and 4R? It's easy. What is 3R? 3 repeats and 4 repeats of the tau. So in case of Pick's disease, there is a spherical inclusion of the tau around the nucleus. In case of you can see that it is the tau which is getting accumulated, correct? But depending upon the location and so basically depending upon the location and the number of tangles, it is causing a spectrum of disease. So what could be general symptoms of the neurodegenerative diseases, which is memory loss, motor deficit because of the stiffness, tremors and dementia and impaired language. So far we find that okay, there is Alzheimer's by the amyloid beta and tau, and alpha synuclein in Parkinson's, then 3R tau R spectrum. Then, but it is not that Alzheimer, the neurodegenerative diseases research has stopped, that we have done everything sorted. No. Why? Very good. Before that,
they take very long time to.
very good too. And one of the very common and most important reason. Yes, that's the future. Yes, the common heterogeneity. So yes. So sometimes so they take one thing is they take asymptomatic when the first symptom appear, and when it is the full blown Alzheimer's, it sometimes in some people the duration is very long. So first thing, why there are considerable, I will say, markers, but why we still need to find a good reliable biomarker. Both reason, accurate and reliable diagnosis, prognosis mainly, and the as you mentioned, the therapeutics. So first, many neurodegenerative conditions have extended preclinical and prodromal period where diagnosis using available symptom based assessment is either not possible or extremely difficult due to the subtle manifestation. Second, yes, the heterogeneity because of the co-pathology and multiple other diseases along with the neurodegeneration. So, what could be the most easy way to find a biomarker and most reliable? So we can look for the DNA for the genetic marker. We can look for the RNA by transcriptomics, and we can look for the proteins. But then which you will choose and why?
Easier in the sense that you would have to put genetic markers, it have to be a whole sequencing.
Sorry, proteins why?
If you have a DNA based or RNA based marker,
Yes.
so the process that will have to go through in order to identify that marker in a suspected patient would be comparatively, I would say, complicated.
No, I will not say. RNA, yes, DNA, no, but that's very complicated explanation. Let's find out the easy one. What is the central dogma? DNA, RNA, protein. So who is the ultimate functional machinery? DNA is not causing the tangling, DNA is not phosphorylating the tau. Who is the culprit? The proteins. I will say the functional machinery. Ultimately the thing is done by proteins. Yes, DNA is very good because it's predestined. Okay, DNA, so predisposition. So we know that there is some mutation in APP and some genes which lead to the accumulation, and that ultimately lead to the tau accumulation and everything. Yes, but in quest for the ultimate functioning person, which is protein, we will look for protein as a biomarker because it's easy to, so we target proteins. You do not target DNA. We target the kinase inhibitors, the trametinib, sunitinib, nilotinib, the kinase inhibitor, senescence inhibitor, the quercetin, dasatinib, all those things. So we target the proteins. So if we found a good biomarker, it will be easy for the, I usually not say diagnosis, I say prognosis. So a good protein biomarker will help in the prognosis and the therapeutic. So we are moving towards the proteomics. What is proteomics? Study of? Very good. Yeah, proteome is like 30,000 proteins. So study of the large scale of protein, large number of proteins, and there are multiple ways. So you could do the qualitative proteomics and you can do the quantitative proteomics. What is difference between qualitative and quantitative?
Qualitative would be identification of a particular protein, and quantitative would definitely be the amount of quantity of protein.
Yes. So she is right. If I do the qualitative proteomics, and I am comparing Alzheimer's versus the healthy person, I can only find out this protein is present and this protein is absent. So but it may not be possible. So but if I do the quantitative proteomics, I can also tell that this protein is, then I can compare and I can tell, okay, this protein is 2 times more in Alzheimer's compared to the normal healthy brain, or 3 times less in Alzheimer compared to the normal healthy brain. So or you can and that part of information of the qualitative will definitely be in the quantitative. If a protein is totally absent in normal but present in the Alzheimer, it will appear, it will give its value, the numerical value in the Alzheimer part, but there will be nothing in the normal healthy part. So the qualitative will give you the protein profiling. So it's profiling. A, B, C, D is present in the Alzheimer, and A, B and C is present in the normal, so D is absent. Just profiling. And when I will say that okay, A is 2 times more in Alzheimer's compared to the normal, it becomes quantitative. And what else you can do in qualitative proteomics? You can characterize the post-translational modifications. But usually we also do the quantification of post-translational modification, and the third interesting part you can do with the qualitative is analysis of protein-protein interaction. Any guess how? Very good. I will say using the affinity chromatography followed by affinity chromatography then mass spectrometry. This is what you, yeah. So it's like but always it's not ligand receptor. If there is a protein and its interacting partners, if you want to find out interacting partner of, let's say, HSP27, heat shock protein 27, so you will precipitate the heat shock 27, just normal affinity immunoprecipitation, very simple sorry, which value? We don't need KD value here, we don't need. So you just need an antibody which binds very nicely with the heat shock protein 27 or any protein of interest which you want to study, and it will precipitate HSP27, let's say for example, and it will also precipitate everything that is at the moment in physical interaction with the HSP27, and then subject to the mass spec. Take the sample to the mass spec. So you, in the mass spec data, you will not get only one protein HSP27, but also HSP27 and maybe 26 other protein which were bind at that time of the precipitation. Correct? So, but yes. No, why it will? Yes, but that's very specific, and that's why we say optimization. Okay, so I said immunoprecipitation in one line, it does not mean that it's very easy. So you have to optimize the concentration, how much antibody. Now first what did I say? The first line, you need an antibody which is very good affinity, which he said the KD. So whenever you buy the antibody, sometimes in the data sheet they give the KD value. So you need a very good antibody which strongly bind, but we do not want that much strong also. So good antibody, and then you have to optimize the concentration of antibody, what amount of antibody, what dilution you are going to use for the precipitation. And yes, no, you are not happy?
Maybe I can answer what he said. Would be effective in case if the
It depends. It depends what whose KD value is more or less. Then it becomes proper chemistry. But just imagine, you will miss only one antigen, one protein, correct? Or am I wrong? Or maybe two. But it's not possible that you miss the 20 interacting partners. Agree or not? You will miss 5, let's say 5. If you do the protein-protein interaction, you press it, although we don't miss, trust me, I do. But max to max, you will miss some interacting partners, but then you are not going to miss everything. You are getting information. And also, if you miss something, then you can go back to in silico, and then you can find out sometime has definitely there is a data where we know the list of interacting partners, and then we always compare, okay, here is my mass spec data, here is the in silico, what does it match? So if you are not happy that okay, this is the actual, this was the very good supposed to be, this is the functional thing. Let's say you are looking for the cancer and you miss the MIC. So and you think that MIC is important for your study, then you can do further experiment to find out that does MIC interact with your protein or something like that. You have to go further, but you will not going to miss information because of the immunoprecipitation. Okay, so moving and quantitative, we got the numerical information. Quantitative proteomics, using quantitative proteomics, what you can find, you can do the molecular mechanism or signaling pathways. You can find out the disease biomarkers or identification of therapeutic targets. How? Let's go one by one. The second point is clear, correct? So you can by doing the quantitative proteomics, you can find out a biomarker, or let's say prognostic marker or diagnostic marker of a disease, something. So Alzheimer's is going to be symptomatic, early onset versus late onset, who is going to be symptomatic, who is not going to be symptomatic, all this you can do. Then what is problem with the first part? You will get a list of 20,000 proteins, and then you have to map it depending upon your particular interest. So if let's say out of 20,000, 500 proteins are significantly upregulated in symptomatic patients, then your further concern is only that 500 proteins, not the 20,000. Don't get attached to the data. So those 500 proteins, and then you will put that 500, you will do the bioinformatics on those 500 proteins to find out in which molecular pathways they are involved. So you will map the signaling pathways, and then you can say that okay, 30 proteins are up in particular XZ pathway, and this is what's supposed to be. So I say that okay, EGFR gives the signal EGFR, MAC, ERK, and then further downstream. So if what in my data, EGFR is up, MAC is up, ERK is up, and HSP27 is up. So this is what theory says, this is what my data says. So why not I will choose this, and I will say okay, this is the pathway of my disease. If okay, and depend, so you always get the significance p value, and then if you identified you have already identified the proteins, you map the signaling pathway, then you can target those proteins, correct? You have not heard the mTOR inhibitors, you have not heard the integrin antibodies for integrin is everolimus, bevacizumab, which is VEGF inhibitors. So yes, if you identify a protein, you are close to identifying the therapeutic target. But how you do the quantitative proteomics? So you can do there are by two ways, labeling and label free. So label free is it's, I will not say easy, it's cost effective. You just count the spectra for each sample, for each protein, not you count, the software will count, but it takes time, and measure the ion intensity and the spectral counting, and then you combine both, and then you say that okay, this is up compared to this, and you have to do that independently for both the control group and the disease in question. On the other side, the labeling is easy, more reliable, faster. So then you can do labeling by two methods. There are two ways, in vivo and in vitro. If you are doing like culture and your research question is treatment versus not treatment, you have a model of Alzheimer's disease, you have a model of some any disease of your interest, then you can have that model versus control. Let's say we know that if there is mutation in APP, it can lead to the Alzheimer's. So we knock out or we induce that mutation in a cell, and so now that become the Alzheimer's model, and then we have the wild type where there is the normal copy of APP is there. So then you culture it, then you use you instead of then in the culture media, our media is normal, while when we do the in vivo labeling, we use heavy and light isotopes, and usually on the lysine, and so N14 and 15. So one we will give to the wild type model, and another we will use to the knockout model, and we will subject to the mass spec, and when the the software will compare itself, it will know that okay, this carbon is heavy isotope, this carbon is light isotope. And this method was discovered by my boss's boss. Then comes the in vitro. In vitro method is more handy when you are doing the tissue proteomics, the blood, plasma, all things. So then you isolate the protein, you do the trypsin digestion, and then you label, again the thing is same, you label those peptides with isotopes. So and then isotopes, they don't get like ITRAC, TMT, tandem mass tag, these are like, just consider them as the barcode. Okay, so let's say we have two groups, Alzheimer's and control, healthy. So I will label the Alzheimer's with barcode A, and the healthy control will be barcode B, and then whenever in the mass spec, it will know that okay, this peptide is barcode A, this peptide is barcode B, but they will be close, correct? Because mass spectrometry, it is the basis of mass is to charge ratio. Those two peptides which are same size but from different samples will be close enough, they will be close, So but the intensity may be different or may not be, but the mass spec will identify, okay, the A is more or B is less, something, they will be close, and it will measure the difference in the intensity. So what samples you can use in case of neurodegenerative diseases? The cerebrospinal fluid, the CSF, which should be the most ideal sample because it reflects the brain thing, okay, it reflects the brain pathology, but it's invasive. I will say it's painful. Anybody has seen the CSF sampling? Human CSF sampling? It's painful. So they take from the lumbar spine and with the needle. Second easiest thing is blood, like plasma or serum. It gives us the information, but it is not exact reflecting the brain pathology, but everything has its pros and cons, so it's less invasive than the CSF. Then the easy one, the saliva, tears, urine, okay, everything is happening, so maybe something correlate with the saliva, tears, urine. Now, apart from liquid biopsy, you can do the proteomics with the tissue sample. How you will get the Alzheimer, let's say Alzheimer patient, human with Alzheimer's, how you will get the sample? Very good. There is no other way, no other way. So the autopsy samples. So whenever, if you will read the paper, I found most of the time autopsy sample only, and nobody is going to, they suffer Alzheimer, Parkinson, I am not going to give you the permission to make a hole in my brain and take one part of the sample. No, sorry, nobody gives. So the only option is autopsy. So you get the brain, then one thing is, next slide, sorry, the whole proteomics, the bulk. You get the affected part, the pathologist will tell you this is the affected part, so the pathologist will give you the sample, you take the sample, process it, but that is called bulk proteomic. If you are rich enough, if you are a rich lab, you want to get more fancy and you want to be the cell specific, so then you can do sorting, micro manipulation, labeling, imaging. So sorting, it's very easy. You make the single cell suspension of your tissue, the tissue, make it single cell suspension, and depending upon which cell proteomics you want to study, you need to know the marker. The only drawback of the FACS is you need to know the marker. So which type of cell you want to do the subject for proteomics, you sort it, and then after that is the same, subject it for the mass spec. So it is the good part is highly pure, it gives you the real answer, but the bad part is cell damage during the processing. There are a lot of processing steps. The microfluidics is again the same thing. It's like these days they are very thin very thin capillary kind of thing, and they subject the cells to you have to make a single cell suspension, and they subject it to that fluidic system, and it allows only one cell at a time to pass, so you will get the single cell suspension. Then the most interesting thing is mass cytometry. Anybody has heard about mass cytometry? So mass cytometry is like fax. It's like fax, but in that case we do not use fluorescent labeled antibody, we use metal tagged antibodies, and that's it. This is the only difference between mass cytometry and flow cytometry. Then the manipulation, micro manipulation, it's easy. Laser capture micro dissection, you decided that you want the core of that plaque. You did not trust the literature. You say everybody is saying that there is a plaque. What if there is something more? So you want to take sample of plaque, and your research question is plaque versus no plaque. So you take sample of the dense core and go slowly, like every 1 millimeter from the core, from 1 mm from core, and then 1 mm for core, and you compare all the three samples, and that you can do by either by only if you have the facility of laser capture micro dissection. So in case of laser capture, you get the sample on a slide which has the PET membrane, the normal glass slide, but that will be the membrane, and on the computer we have the image. We say we want this part, automatically the needle will go and take the sample from that part. Another thing is micropipette aspiration, that anybody, we all do. I say that macro dissection because if we are not rich enough, but still we have the sample, so we go to the pathologist, we say please mark the core versus not core, and then we take that part with the fine needle or the tiniest pipette tip. The another thing is labeling. So we label with the particular enzymes, like or proximity labeling, like we label that, okay, this is, now we take the sample, make it single cell suspension, we label that particular cells and then subject for the mass spec. Then the last thing is mass spatial, it's spatial proteomics. I do not say proteomics because till date we are not doing full proteomics on the spatial, so but still people say it's proteomics because we are moving towards. So what is spatial proteomics? Have you ever heard of spatial transcriptomics? No, it happens on the slide, the histology slide. So when we get you process the bulk tissue, you got the proteomic data, but you do not know that which protein is coming from which cell. Perfect. Then you did the single cell suspension, you separated the cell and you did the proteomics, you got the information of proteins from the cell, but you don't know where this cell was histologically, what was the location, correct? But the spatial proteomics is giving us the advantage of it does on the slide. So from each cell it gives us a colored photo, it from each cell it will give us it will say, okay, this is oligodendrocyte and this is the proteome of oligodendrocyte. So it will tell me, okay, you will get very fancy colorful colorful image of the slide, usually what we get from the histology slide, either brown color, brown and blue, the IHC, or 3, 4, 5, maximum 6 to 8 colors on the, or if we are doing multiplexing of IHC, 6 or 8 colors of the immunofluorescence, sometimes 40 if someone is going to be more picky, but the spatial transcriptomics give you each cell type. So it will say this is the endothelial cell and is this the proteome, and there are 20 endothelial cells, there are 400 oligodendron, there are 400 Purkinje fibers, and this is the proteomics, and it will be colored. So it gives pseudo color to the each cell, so you get beautiful color map and parallelly a data. So you got the sample, and based on your research question and I say affordability, what technique you can follow. Now what proteomics you are going to do? The whole proteomics is two things, the bulk thing and then just proteome. You process it and you get it. But what is the PTM? The post translational modification. So it is not necessary before that. How many type of post translational modifications are there? Name 2, 3. Methylation, phosphorylation, glycosylation, ubiquitination, and acetylation, sumoylation, blah blah blah. List is long, but phosphorylation is the most common one, and it is not necessary. So if you find a kinase which is overexpressed, so what is requirement of kinase? Or it has to be active. So kinase, so let's say I am in the department, I am teacher, but I am not teaching. Okay, I am not teaching. So what I am doing? Nothing. So I am there or I am not there, it does not matter. So that's the position of a kinase. So if a kinase is overexpressed, let's say there are 20 copies but not active. So usually it is not always expression that matters, it is the activation also that matters. So once it gets phosphorylated, it gets active. Correct? Agree? Everybody agree? Nobody disagree? Not always. The site specific. Very good. Then, who will give you this information? The mass spec, the proteomics. So you can do again. So let's say the phosphorylation. We want to study the phosphorylation, the most common one. And phosphorylation happens on how many sites? Where? What are the sites? Sorry? Very good. Serine. Someone said something. Threonine. And the most least abundant but most important? Yes, tyrosine. And these days they have discovered that histidine also gets phosphorylated. But let's ignore histidine. So tyrosine, serine and threonine, these are the most commonly phosphorylated. And yes, you are right, depending upon the site it gets activated or deactivated. or inhibited. So how we get the phosphorylation? So when we do the proteomics of phosphorylated peptides, we say that phosphoproteomics. Okay, so phosphoproteomic, glycoproteomics, that's it. I have never heard sumoproteomics. So but then ubiquitome and acetylome, so they then the proteomic, they leave the proteomic part and keep the ome part. So phosphoproteomic, glycoproteomic. So how you get the phosphorylated peptides? You got the proteins, you got the sample, extracted the protein, and now you have extracted the protein, digested with the trypsin. Now what? You have 20,000 peptides, not everything is phosphorylated. So, and I want only phosphopeptides with phosphorylation. Yes, yes, and then what? Yes, you are right. Yes, go further in the right direction. No, we don't do ion exchange. Ion exchange is the part of mass spec, but we don't do that for enrichment. Okay. Yes, affinity. Yes. So how? Yes, she is 70% right. I am very happy. So because the peptide which are phosphorylated has the 2 negative charge, so there are multiple affinity chromatography methods to do the enrichment of phosphopeptides. So you can do with any metal which has 2 positive charge. So nickel, Ni 2 positive, Fe2 positive, iron, or IMEC. Someone presented the thing IMEC, immobilized metal affinity chromatography. So then initially people say, okay, nickel 2 positive, very good, we found the method. So they did not get the enough number of phosphopeptides. Then someone says, okay, Fe2 positive, let's do Fe2 positive. They did not get enough number. Then someone combined the metals, Ni, nickel and Fe2 positive both. So instead of saying metal, so they immobilized metal affinity chromatography and they get better results. What I do is titanium dioxide method, so TiO2 positive. So then titanium dioxide, it's easy, it's very easy. You have to get the beads, incubate your peptides with the bead, elute it, done, sorted. So and this process is called enrichment. So we do the phosphopeptide enrichment, we do the glycopeptide enrichment. So when we take out something exclusive which is of less stoichiometry from the mass thing, we say enrichment. So this is called phosphopeptide enrichment using your choice, nickel, IMAC, titanium dioxide. But the drawback is you get very less number of phosphotyrosine, because the most abundant is serine. The ratio is 92 to 8 to 0.1 in normal human normal condition. So tyrosine is 1 to 0.1% in normal physiological condition. So we do not get phosphotyrosine enough to draw some conclusion. Then what we do? Is there any way we can do something? Then we do the immunoprecipitation. Now we know that we want the phosphotyrosine sink, so we get the antibody which detect the tyrosine. So we get the anti-tyrosine antibody, and in the world there is only one antibody that everybody use. So the phosphotyrosine antibody, and do the immunoprecipitation, and you will be surprised to see that you will not believe that okay, this much number of tyrosine were phosphorylated in my sample. So yes, enrichment, immunoprecipitation if the stoichiometry is further low. Then, so that was the whole proteomics. Then the modifications. Then is the aggregated, if you want to study the plaque, the infarction, the previous, you just need to take that part and subject it to the mass spectrometry. Spatial we discussed. And how do you study the turnover? I just told. So proteins get degraded when they get no, not because of us. They get degraded on the temperature problem when we do physiologically. What is the modification that guide the cell that okay, this guy is ready to die? No, ubiquitination. Don't talk miRNA to me. I'm not.
So what is the name of the antibody, antityrosine antibody?
PTYR 1000. So it's like phosphotyrosine 1000. It's very costly, super duper costly. But yes, if you want, you can do. But it's worth trying. If you think of tyrosine phosphoproteome, it's worth trying. If anybody wants, I have it. At least I'm rich on something. So briefly, you do the proteomics, you find out the disease biomarker, and by mapping the proteins to the pathway, you find out the mechanism, and then you can target. And also, why the first problem we discuss is the heterogeneity. So my proteome could be different from the another person's proteome. So because definitely there is a reason why one person is developing at 20, why one person is developing the symptoms at 40, another at 60, and so there is definitely difference in the proteome. So that's where comes the role of personalized medicine. So if you, this is very, we all say this, we all are trying this, but this is very dream situation, that whenever we get the sample, it's like for the personalized medicine, things should be happening in the pathology, correct? Sample they perform mass spectrometry on every sample, and they say okay, this pathway is in this patient, this pathway is in this patient, and then comes the problem of clinician. Okay, he has to read every report and say okay, you are going to get this, you are going to get this, you are going to get this. But this is our dream. We are working for the humanity. Back to the phosphorylation. What are the functions that are regulated by phosphorylation? It's not just activation of kinases. It is involved in the molecular association, enzyme activities, intracellular localization. Agree? It's very important one. And then crosstalk. When something get phosphorylated, then something get acetylated, or if something is acetylated, it does not get phosphorylated. So the crosstalk. Protein degradation, when something is phosphorylated, then it gets deactivated and it does not lead XYZ protein to get ubiquitinated, so the protein is not degraded. So it controls the protein degradation, protein stabilization, conformation, and processing and receptor cycling. And the processes regulated by phosphorylation is there are vast functions including neural plasticity, cell growth and adhesion, differentiation, everything. Very briefly, this is what I do. So you get the sample, just omit my part. So and you can have any sample, your Alzheimer's, normal brain. You take the protein extraction with the help of lysis buffer, and then we do the trypsin digestion, because we want the we in the mass spec we identify the peptides, and then we label these peptides with the tags in an easy language, the barcode, and then we pool it. So this is one of the advantage of labeling. So I started with 3 samples and I end up with 1. So from this step onward I have 1 sample to process, but and the cost is this thing, the TMT tag. This is the costly affair, but then after that it makes the life easy. So if you are doing the enrichment or even the mass spec, if you are doing normal proteomics, go directly from here to here. The sample is ready, labeled, subject it to the mass spec. One tube is going for the mass spec. Imagine you were doing the label free, so you have 3 samples will be going, so the cost will be 3 times, and it will take more times to process, it will take more time to analyze. And also if you have planned something, the PTM study, you have to do enrichment, now you have to do the 3 enrichment in 3 tubes. And what if your antibody is PTYR 1000, you have to take 3 aliquots. So everything has its pros and cons. It's costly, not very costly, it's costly, but then it makes life easy. After this, 1 tube enrichment, 1 tube subject to mass spec. Mass spec is easy peasy, it will give you the quantification, and this is called relative quantification, and then that's it. This was the process. This I bring for you. So our dear spatial proteomics has been said as the method of year 2024. So in the Nature methods, so this is the fancy thing, and everybody is doing spatial, whatever they can do, spatial transcriptomics, spatial genomics, spatial proteomics. Everybody, now we don't want the proteins in the tube, we want to identify protein in the tissue itself. So you can see, so they have labeled hippocampus, so this, the colorful thing is hippocampus, and they of Alzheimer's, I struggle to find out this good one thing, and then different cell types. So neurons are orange, where is orange? Orange, magenta, green, astrocyte, everything. But the interesting thing is this one, the plaque. So they manage to color everything. Then they have the data. So they have the data of this purple the proteomics data from the purple thing, proteomics data from this orange thing, proteomics data from the yellow thing. So now they know this is the arrangement. Plaque is here, which is between astrocyte and which there is a magenta thing is neuron. So between neuron and the astrocytes are the plaques, okay, but this information you will not get if you crush the whole tissue, correct? And it's always nature is more smart than us. We have reached to this close, but we say that okay, here is plaque, but who decided the plaque will be here? There must be something, and that something information we are getting closer to and closer. So that something information can come only by spatial transcriptomics, because this will tell me that okay, these endothelial cells, these astrocytes, when they get changes in XY protein, they lead to the accumulation of plaques. Then another fancy thing is single cell proteomics. You have heard of single cell transcriptomics, single cell genomics? No? So it's okay, everything is same. They have just, instead of doing the bulk, we are doing with the single cell. And doing the single cell, there are multiple ways. So one thing is you can do the FACS, but nobody like it. The fluidic method, the microfluidic is yes. So we get the machine which has the plates with the microfluid, and it allows only one cell in each well. So yes, this one. So this is the, you will be having, let's say 96 well, and each well will have further wells, and the well will contain, each micro well will contain only one cell, and then each micro well will have its own barcode. After that, barcoding is done, so each cell is coded. Now I know that cell 1 is whatever is it is, cell 1, cell 111, cell 2, cell 2 2.3. So every cell is coded. Then after that it's same. We perform the proteomics, and then we know based on the biomarker for that particular cell, okay, this proteomic signature is coming from the glial cell, this proteomic signature is coming from the astrocyte, and that's it. Now to change the flavor, the real data. How many people are working on neurodegenerative diseases? I know, except me, most of you. Or everybody is working? All people are Alzheimer, Parkinson's. Very good. Are you aware of this consortium? No. So they made a global, there is a global neurodegenerative proteomic consortium. I know, I am not the, so this is a part of, are you aware of Human Proteome Atlas, Human Protein Atlas, HPA? No. HUPO, Human Proteome Organization. You know HUPO? Okay, so there is Human Proteome Organization, and which have centers across the world. And in India there is only one center in Mumbai, Dr. Sanjay Nawani. They are pathologists, and or anybody, one thing they are designating, anybody, I can submit my IHC or proteome data, that here is data, you use it for your thing. So they made the Human Protein Atlas, so that contains all the protein information, the real information, the immunohistochemistry. Now they have made the cell atlas, this. So finally the same people have made the global neurodegenerative proteomic consortium. So this include specifically Alzheimer's, Parkinson's, and the lateral sclerosis, and this dementia, frontotemporal dementia, and aging. And this is from the 23 cohort, means 23 centers across the world, and look at the sample size. 31,083 samples they have processed, and I'm getting too much time. Please, so and then for these samples we will go on till someone comes. So this is interesting. So they have performed, so in short they have large cohort of sample representing the whole world. So they have sampled definitely from Asia. I have not read there India is there or not, but Asia is there. Asia, UK, Europe, everybody. And then they measured 250 million proteins, 250 millions. And what they have found, so everybody is familiar with this, the volcano plot thing. And so they compare Alzheimer's versus normal control, Parkinson's with control, and what they, so these are the upregulated proteins, these are the downregulated proteins, and in the same case. So if you are aware, you know better than me the neurodegeneration. Alzheimer's, it is acetylcholinesterase, and so these are the list of genes which are up, and same case upregulated in Parkinson's, and interestingly there is nothing upregulated in frontotemporal dementia, it's downregulated, and yes, in lateral sclerosis. Then you perform the mass spectrometry, you decided to work on neurodegeneration, you got the tissue cell, whatever you got, you perform the mass spec, you got the data, you converted data into this fold change thing. Now you know that okay, these proteins are up, these are down, what next? What you can do with these? This is not enough until unless you say that okay, this protein is involved in this process via this pathway. So you have to find out the molecular function, the pathway analysis, and protein-protein interaction, and many more things. But these are the main thing. How you do this? So there are two methods. One thing is paid softwares, and another thing is freely available. So anybody aware of this? Ingenuity pathway analysis. So this is paid software. It's from Kaizen and it's paid software. It gives the free trial option if you have the institutional ID. So, but it gives you very good information. It can analyze your upstream regulators, signaling pathways, and it can tell you that your protein is involved in which other diseases, so you can correlate the pathology, and it can compare the expression pattern, mechanism, everything. And this is the real data, this is my data. So, and it is and it can, so the red thing are overexpressed and green thing are underexpressed. So it will map what is the location as well. So I know that these protein are membranous, these are cytoplasmic, up and down, what is up of this, what is down of this. So I can think about the mechanism. But that's not the end of the world. If something is paid, there are free software. This is totally free, you can enjoy. You have to download this. So the FunRich, I love this software. It gives plenty of information. It gives you the gene ontology, biological pathway, protein domain, site of expression. Like it will tell you ki, okay, you found this protein in Alzheimer's, that's your data. But the software will tell you, okay, this could be the microglial cell, this could be the astrocyte, this could be the lymphocyte. So they give you a number and the p-value. So we choose the first criteria in biology is the p-value. Something has to be significant, so based on significance we select, and it gives you the transcription factor. So difference between this one and the free one, software does for you. You say click, just click the button and it will say okay, I want the upstream upregulator of the TGF1, so from your data it will do this and it will give you beautiful diagram like this. Here look, it is giving all the information. It will tell you the pathway, it will tell you the transcription factor. So if you know the transcription factor, if there are 10 proteins which are overexpressed and you know the transcription factor, so you definitely know that this is the transcription factor, it is leading to the overexpression of these genes, and then you can go back to the literature that this transcription factor get activated when there is activation of, for example, NF kappa be activated when GSK3 beta is phosphorylated, correct? So when it's phosphorylated, NF kappa B is activated, it goes to the nucleus and do the transcription. So you can go, you have to go back and forth and you have to use your time. I will not say waste, but use your time and the intellect. I love this. Then there is, this is very easy peasy tool, it's CynyGo, it's online web version. You have to do nothing, just paste your list here and it will give you so much information. It will give you the pathway involved, and you people have heard of KEGG pathway mapping. You need not to do this, this guy will do for you. So it will give you the pathway, this, and then you select the pathway which one you want to map. So it will give you, okay, this is the pathway, and it will do the color thing. Okay, in this pathway, let's say P53 pathway come up in your thing, and all the red one are upregulated and they are in your, so out of 20, 10 are in your data. Then usually we do the string. Have you ever heard of string? So we do the string analysis for protein interaction. So this software even cut that. Okay, don't go anywhere. If it's like mall, if you go to the CynyGo mall, everything you can do in the same place. So you can do the, find the pathway, you can do the mapping of the pathway, you can do the protein interaction, and which I love most interesting is it gives the genomic localization. So you can go back to the gene. So now what this information gives, that, so this is the enriched, means whatever this particular, so this figure says that chromosome 20 this overexpressed, so mostly overexpressed gene is on chromosome 21. If we zoom, it gives exact location. Then you can, then depending upon your research question, you can go back and say, okay, I want to do the gene amplification, like I want to do the PCR, that what if there is copy number variation. So what if the problem is not at the transcription and translational level, but it's at the genetic level, there is duplication of this part, correct? So it gives you the genome location also. Back to the continuity of this data, which is your Alzheimer's thing. So based on all those, the pathway analysis, they did the, after doing the volcano plot and expression, they did the pathway analysis, and they find out that in Alzheimer's, this is the glucose metabolism and Rab granulation and endosomal sorting, these pathways are up, while in Parkinson's contains a lot of GTPases. So it's full of Rho GTPases pathways, and the temporal dementia has protein translational, protein phosphorylation, IGF signaling, and so doing all those mechanisms which I explained. They mapped. Don't worry, this is just a different way of presenting the data. We say this is the bubble map. So you can do by whichever. Another interesting thing you can do with your proteomic data, so they have predicted the difference between the chronological age and the predicted age. So based on proteomics, they correlated that based on this proteomic, what is the does the proteomic age correlate with the chronicle age, and they found that yes, it correlates, and using this as a baseline data, this was the baseline, this was their previous mechanism. What is the information that usually, what do we think? Alzheimer's brain is the problem, but they have shown that cognitive brain mostly correlated. So there is in Alzheimer's the gap between the chronological and the disease age is more in the cognitive brain, the bold ones, the artery, the liver and intestine. I have not imagined about this information. And then in case of this dementia, it's cognition brain and artery. Then in the Parkinson's, muscle is expected, but lungs, maybe because ultimately it has to function in a muscular way, but Alzheimer's was totally surprised for me. So proteomics can give you the pathway mechanism, it can tell you. So what did information we get from this? That nobody knows that this lead to this or this lead to this. So the paper quote that we don't know its causation or consequence. So you have the arena open. So another study they did the plasma proteomics of, so this study is comparing symptomatic and asymptomatic patients, and they found yes, ACHA is there, but SPC25. And when they correlated with the clinical parameter, these number of proteins correlated. The red one are over, and the more positive correlated, and the blue one are negatively correlated. The interesting thing is here. So these 20 proteins, the MIA and called 10A1, these two proteins are involved in cytoskeletal and extracellular matrix, and they correlated with the symptomatic progression of the disease. So if there is progress, if the with progression they decrease. And then they correlated, they find out the pathway. Nervous system we expected, but the ECM and the lipid pathway may be new for this. Then what do you, so it's the neurodegenerative diseases, correct? So what they are having, cognition affected, memory affected, so must be something which is common. So this study find out that 836 protein are exclusively to Alzheimer's, and 673 are exclusive to Parkinson's, and only 270 are for this dementia frontotemporal, while these are the overlapping ones. So between Parkinson's and Alzheimer's there are 1600 proteins overlapping, and between all three there are 996 proteins overlapping. Then you can go back and you can do the pathway mapping. You put as a homework you can do, as a fun, you go to this paper, take these 996 proteins or 16 and put in the shiny go and see the magic. Then you will find out that okay, this pathway is common to everyone and this pathway is specific, and thus this is the mapping like common and the specific one. And the last study, this is my favorite study. So they have done both proteomics and phosphoproteomics on the same samples, and this is like principal component analysis. They say that okay, this is like based on the age. Now people started correlating that okay, with age memory decreases, with Alzheimer's memory decreases. So what if we found the gene which is like with the aging, and what is common between aging and Alzheimer's? So if we found something for the aging, we can find the, can we target the same thing in Alzheimer's? So based on this question, they divided the animals. So this is animal study, everything else was human. So adult, mid-age, which is green, and the old one, and you will find out. So this is the proteomic data, and these are the clusters. They clustered like up down on the, and you will see that these clusters, the proteins are overexpressed in old, and these is like, so only one case there is underexpression. And what is, and this is the pathway. So signaling to the GTPase goes down as with the aging. So we can hypothesize that maybe this is something happening similar in the Alzheimer's. And what else is happening? Yeah, what is going up? Neuroinflammation. So inflammation, all this goes up with the aging. So we can hypothesize that maybe something like this is happening. Then based on this beautiful thing is kinome tree, and it represent, every branch represent the kinases, human kinome. These are 500s. So based on phosphoproteomic data, because you got the phosphopeptides, you go back to the in silico analysis, and you can deduce that okay, this phosphopeptide is phosphorylated by the kinase, this kinase. So from that information you can find out the kinases which are involved in your disease question. So here they have said that these kinases are involved in the aging. This is total they identified. And this is the phosphoproteomic data, and we can see that the overexpressed one are the yellow one and the old one, so the gray. We have to look at this, the gray thing, and gray thing is glutamatergic synapse, synaptic, so all this we found in the Alzheimer's. Agree? So we can correlate that something which is happening with the aging maybe similar is happening in the Alzheimer's. Yes, they are definitely more smart than me. So they find out the phosphorylated, so they know, we know that these phosphopeptides are correlated with neurodegeneration, and these phosphosites are cellular senescence. The easy peasy one is the tau, MAPT is tau, correct? And all tau is hyperphosphorylated in the old mice. So now do you agree that with aging tau is getting phosphorylated, and that we know that with aging memory goes down, so maybe is a good model for the Alzheimer's, or at least it can generate a good hypothesis. So this is the kinome of aging. These are the kinases which are associated with aging. In 2 words, what information you get, that most of the kinases are from the CAMK family or AGC family or STE family. So you cannot target tyrosine kinase inhibitor. So nilotinib, sunitinib, dasatinib, blah 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 blah, any IB is not going to work on the Alzheimer's patient. So you have to find out something from the CAMK. You either the inhibitor is present, you can try. If it is not, you have to develop. So we know that the CAMK, or at least you get the filtered information that these are the kinases I have to find out, and this is what I do. And the same study showed that the changes are kinome changes are involved in the neuroinflammation by cytokine signaling, neuroinflammation, and the clustering CD molecules. This is what we are doing with the Rajat sir. So he is focusing on the neuroinflammation part of the Alzheimer's. This study shows that with aging there is breach of the blood-brain barrier, and that can lead to the cognitive impairment. That can lead to the cognitive impairment, and when they, and the one of the most common, so how they say the breach is there, because they compare the CSF to the plasma ratio of the protein. So they did the CSF proteomics, they did the plasma proteomics, then they find out that which proteins have higher ratio. The CSF to the plasma ratio was high, and fibrinogen is one of the protein which correlate with the aging and the cognition. With severity of cognition, it's not very high, but it's high. MMSE is the test for cognition. This is the study. It's like ataxia related. It's the neurodegeneration. In this disease, the Purkinje fiber neurons, they get degraded. One of my colleague in UK have done this work. So and they know that this gene when it get mutated, it leads to the so look in the wild type, the green thing is vimentin. So in wild type, when SACS gene is normal, the fibers are nicely arranged, and when it get mutated, it is like there is no arrangement, it's accumulation. So that leads to the movement disorder. And when they did the proteomics and the phosphoproteomics, they found overexpression of tau and overphosphorylation of tau. So one more thing added to the tauopathies. And when they did the kinome mapping, these are the kinases they have given to, like the red one is overactivated, and the specific tyrosine 262 was, so because when they did the kinase enrichment, they find out that BRSK2 kinase. Nobody ever heard of BRSK. We nobody bothered to BRSK2, but this is the most significantly enriched. So when they validated, if someone will ask me to do the kinase, I will choose very fancy one, even the EGFR, AKT, ERK, MACPAC, but nobody goes to these one. And they surprisingly they found the knockout cells have overexpressed BRSK2 and hyperphosphorylation of tau 262. And then they did the surface proteome. So far you did the bulk proteome, you did the single cell level proteome. Now you can go to the subcellular proteome. So this, when I say surface one, this is the membrane. So there are two ways to doing the membrane proteome. From the bulk, using the bioinformatics, you choose the only pick the membrane ones which are bioinformatically localized, or you do the real experiment. You do the immunoprecipitation with the biotin, correct? And so immunoprecipitation is very handy in the proteomic field, and they found these genes and the pathway which were related to the Golgi membrane, extracellular exosome, and these enriched in the ataxia disorder. I think that's it, and this is what I do. I don't work on the neurodegeneration, I work on neuroendocrine, and I have done the phosphoproteomics of non-functioning pituitary tumors, and we found significant difference between the recurrent and invasive. Usually people say that if it is invasive it will recur, but there is molecular difference as well, and is like normal to the physiological thing. We found serine as one of the most and tyrosine less, because we did not do the tyrosine PTYR 1000 precipitation, and these are the list of phosphopeptide which are significantly upregulated in the recurrent one. The red thing shows, and volcano shows that beta catenin, ASHD and ITH2. The drawback of phosphoproteomics is you get mass spec give the information, but then you do not get the antibody. So for validation, either you do the mass spec of different type or you leave it. So I did, out of 3 I did only this one for the validation. I also have done the proteomics of the same data. It's always ideal to do proteomics and phosphoproteomics or whatever PTM you want to do on the same sample. Then you can compare the thing that okay, it is not overexpressed but it is phosphorylated, it is not overexpressed but it is glycolated. So I found a different set of pathways in invasive and recurrent, and there was different upstream regulator for both, which clinical people says that okay, if it is invasive it is recurrent. My answer is no. They slightly disagree with me. And then what? So, and this is the information I get from the Ingenuity pathway, and we say that okay, it's the integrin linked kinase is involved, and I validated with the immunohistochemistry. I'm not going to bore you with this. This I already showed, and this is the PhD student I'm co-supervising. So we also did the phosphoproteome. This is the proteomics in another type of endocrine tumor, the sparsely granulated tumors, and we found the overexpressed protein which are labeled, and then we deduced. So this is whole thing is bioinformatics. The theory is we found TNIP overexpressed and majority of our proteins were underexpressed. TNIP is 111 times overexpressed. So we said, with the bioinformatics we deduced the what are the transcription factors involved in the underexpression of protein, and then we find out that TNIP1 is regulating one of the topmost enriched. So TNIP down regulate ELK1, and that may be the reason of down expression of all these 200 proteins. And this is the phosphoproteomics, and thank you. Any questions? Ask anything on proteomics. No? You want to say something? Go on. Any silly question, if anything comes to your mind, maybe I don't know the answer, but you can ask. Anything, shoot for the proteomics. So you know, you can think about experiment, what you learn, what all you can do with the proteomics, the pathways, the mapping, going back and forth from protein to genetic and everything, all you can do. Great, thank you. Okay.